Well, hey there, everyone. Welcome to the Fungi Oracles. My name is Ash. I am your host. This is our very first episode, and I'm so excited that you're all here with me and that you've all chosen to listen to this new podcast about fungi. This whole podcast will be about the kingdom fungi and all different types of fungal species. And there's going to be a special way that we are exploring these species together, which I'll explain here in a sec. But before we do that, I would like to just introduce myself a little bit more. Like I said, my name is Ash. I am a teacher by day, and by night, I'm an avid reader. I really love to work out, and I love spending my time listening to podcasts that I enjoy. And when I was when I have been looking for different fungus or fungi podcasts to listen to, I found some really great ones, but none of them are quite scratching the itch that I need to be itched. So I've decided that I am just going to make the podcast that I want to listen to. And that what that podcast is deep diving into specific fungal species, talking about their biology, talking about their physiology, and talking about how they might impact the human body, either in a deadly, nutritious, or somewhere in between way. So that's what we're going to do. We are going to look at individual mushroom species, fungal species, I should say, and we're going to talk about their anatomical structure, their niche, their evolution. We'll talk about their life cycle. We'll talk about how they impact humans or how they impact other species around them. And that's really the purpose. Another purpose is to keep this pretty darn simple. I want to keep this information accessible to everyone. I did get my bachelor's degree in biology, but I want this podcast to be far reaching and I want everyone to enjoy it. So we will take some time, especially here at the beginning while we're getting to know each other, like deep diving into these, what I'm going to call Biology 101 words, and we'll define them together so we have a nice baseline as we go deeper into other episodes. So hopefully over time, the information can go more into depth, but especially as we're starting out, we're going to take it real slow. It's going to be Biology 101 coded so that this information is accessible to everyone. Now, the way that we're going to be choosing each fungal species, and this is why this podcast is coined the Fungi Oracles, is I have this tarot deck, and you're already a little confused because I told you it's a podcast about fungi, and now I'm bringing up tarot. Tarot is a nice little side hobby that I have, and my favorite deck is called Midnight Magic, a tarot deck of mushrooms. It's by Sarah Richard, and at the end of each episode, we'll go to this tarot deck together, we'll shuffle it up a bit, and we will let Midnight Magic choose our next fungus species to research and to talk about and to learn. Now, today is going to be a little bit different because this is our very first episode, and I wanted to start off with a mushroom that is notorious for being very deadly, aka our death cap. So when we start here in a couple of minutes, we're going to start with the death cap. But from here on out, we will be going to lovely Midnight Magic to find out where we're going or where we'll be headed here in a couple of weeks. Before we start learning about the death cap today, I have a couple of different people I want to shout out. First is my lovely dear friend, Kat. Kat, I asked her to be a part of this podcast with me, and I immediately felt bad about it because she is in the depths of studying for the MCAT and applying for med school, which seems much more important than what I'm doing here. Not that this isn't important, but hopefully you get what I mean. However, Kat did come up with the name the Fungi Oracles. So she is here with us all the time, and she also created the artwork that is the cover photo for this podcast and is on all of our socials. And if you're wondering what those socials are, you can go to at the Fungi Oracles, all lowercase, on Instagram, and you can go to at the Fungi Oracles, all lowercase, on TikTok to find us. And that artwork that you're seeing there on the cover photos is made by Kat. She is so talented, and I'm so glad that it turned out the way that it did and speaks to what I want this podcast to be. The second person that I want to shout out is my dear friend and lovely sister-in-law, Sarah. I also brought up this idea to Sarah and soft launched it as a team effort. And again, immediately felt bad about it because my friends have big things going on. And my sister-in-law just had her first baby, my first nephew, and she has a full-time job right now making sure that my lovely nephew stays alive. So I didn't think that it would be fair to ask her to take on this commitment with me at this time, but it's my hope and my dream that both Kat and Sarah can come and do a couple of episodes with us every now and then because they are both resident lovers of fungi, just like we all are. So with all of that being said, we're going to go ahead and wrap up our introductions. And after this first break, we will take our first step into the fungi oracles, and we will be learning about the death cap together. I'll see you in a sec. Do you like books? Books of all kind, whether they be romance or fantasy or the latest historical fiction? If so, I have the perfect place for you. On Instagram, if you go to Lit Latte and Lawyer Live, you will find my dear friend Olivia and all of her wonderful and lovely book recommendations. The book recommendations come from the highest esteemed authors to indie authors and everything in between. Go to Lit Latte and Lawyer Live on Instagram for your next book recommendation and cute little book find. Now let's get back to the show. Our story begins with the death cap, also known as Amanita phalloides. So it's a part of the genus called Amanita. Now, this genus, we'll talk about the genus kind of widespread here for a little bit and talk about what this means. And then each episode, we're going to take a similar structure. So we'll start off by talking about its niche, how does it interact with the ecosystem around it, what is its anatomical structure, and then we'll go into how this fungal species evolved, where did it evolve from, how long ago. Then we'll talk about where we find this on our planet today, which is kind of like its niche, but sometimes human impacts can spread these mushrooms and these fungal species far and wide. We'll also talk about the life cycle of the fungal species and how it reproduces. And then we'll end each episode with how these fungal species either directly impact humans or if we don't come into contact with them a lot, then how they are directly impacting other species in its environment. So we'll look at that from really like a physiological standpoint. We might get into a little bit of history, but that will just be for fun at the very end. But today we're starting with Amanita phalloides, and I want to talk to you a little bit about this genus Amanita. For one, they're very, very toxic. All of them, death cap being the most so. We'll talk about those toxins more later. First, I want to hit on the fact that these are what we call mycorrhiza fungus. What this mycorrhiza word means is that they have these symbiotic relationships with usually and particularly trees. Now, depending on which Amanita species, and there are 600 of them, they're going to form relationships with different trees and different species. But what that relationship looks like is they are going to basically swap nutrients with each other. What the fungi is offering in water, the plant or the tree will be offering in carbohydrates. So the tree or the plant is able to do that through the process of photosynthesis. But our lovely Amanita friends aren't able to photosynthesize, and they've also lost the ability to decompose along the way. So they need those carbohydrates to stay alive, and they're getting a stark majority of those carbohydrates from their tree friends. So you'll see them colonized around trees a lot of the time because those trees are giving the mushrooms or the mycelium nutrients. And where they're gaining nutrients, they're giving away some water to the trees. So it's a really beautiful symbiotic mutualistic relationship that these have. Now, if we look at the anatomical structure of a death cap specifically, we know that they have these lovely symbiotic relationships with trees, so we'll find them around trees. But a death cap is a mushroom. So, in a mushroom, since this is our first episode, we're going to talk about the different structures of a mushroom, and we'll also talk about how these structures look specifically in a death cap. In a mushroom, we have typically 7 different structures, like macro structures that we can see. And 2 of those are going to be underneath the ground. And that's where a lot of the magic is happening. So I'm going to hit on those first. These 2 structures underground form what we know as the mycelium network. And these mycelium networks are going to span for sometimes miles and miles and miles. And it's has 2 different structures in it. We have hyphae. The hyphae are going to be these little thread-like, arm-like structures that are reaching out all over the soil. And then on the tips of those hyphae, we have what are called nodes, more specifically mycorrhizal nodes. And those nodes are responsible for actually forming that relationship with the tree. So the nodes are going to penetrate the roots of trees so that the tree and the mycelium can talk to each other a little bit, and so that they can swap those nutrients that we were just talking about. And like I said, these mycelium networks are spanning for miles, and they can live for decades. They're mighty cool, and they communicate with all species underground. It's really, really fascinating. Now, coming up from these mycelium networks is when we actually have the fruiting bodies of death caps. And the fruiting body is what we have coined mushrooms. On a mushroom, we have 5 different structures that I'm going to talk about. And I'm going to start at the base. At the very bottom, we have what's called a vulva. The vulva can look a couple of different ways, and sometimes we can't even see it because it's underground. But it's basically this protective structure, sometimes looks like a cup hugging the stem of the mushroom, protecting the stem, especially while it's growing and maturing. From the vulva, we have our next structure, which is the stem. Just like a stem of the plant, it's going to be this structural support as we go up on this organism. And then from the stem, if you look underneath a mushroom, if you're ever laying on the ground enjoying your whimsical day in the forest, and you look underneath, what you're going to see are these membrane structures. And those membrane structures are called the gills. Now, depending on the mushroom, the gills can reach all the way to the stem, or they can leave a hollow area next to the stem. But these gills, their main job is to produce our next structure, which is going to be spores. Spores, we cannot see these units as individuals, but a typical mushroom is producing millions of spores, and these millions of spores together basically create this paste or this dust. And we can see that dust, so we can see the color of the spores when all of these millions of spores are together. What the spores are the reproductive structures of our mushrooms, and they are going to disperse through the wind or disperse through other animals that might be carrying them so that they can go and create new mycelium network. Last but not least, protecting the gills and protecting the spores, we have the cap. And the cap is the very top of the mushroom. It's this very boopable part of the mushroom. Maybe don't boop them. At the very least, boop with caution. But the cap it's going to be different colors and different shapes depending on our mushroom species. Now, if we look at the death cap specifically, the death cap has a stem that is going to be 5 to 15 centimeters in height, and then the cap is also 5 to 15 centimeters in diameter. So they're these proportionate little lads. When we think about the color, death caps are going to be like a white, cream, sometimes yellow or pale green color, and they don't have any warts on them. So their cap is going to be quite smooth. They look a lot like an edible mushroom that you would get from the grocery store, which is kind of the problem. And while that may be a problem for us, it is actually an advantage for these death caps. They've evolved to mimic these edible mushrooms that we might forage or that other animals might eat. How did it evolve to have these specific traits? Great question. Let's talk about it. Before we talk about the evolution of death caps specifically, I wanted to shout out the evolution of fungus as a whole because this happened 1.5 billion with a B years ago. And that in and of itself is so cool. It puts them on Earth before both plants and animals, but that's all fungus. How that happened is a single-celled protozoa from 1.5 billion years ago evolved into what we now know as fungal cells. Now, these cells are eukaryotic, which means that they all have a nucleus. We are eukaryotic organisms, plants are eukaryotic organisms, but it does make them different than something like bacteria, because bacteria would be a prokaryotic kingdom, and those do not have a nucleus. But we're going to fast forward quite a bit. We're going to fast forward all the way to 50 million years ago because this is when death caps specifically started to evolve and the genus Amanita started to kind of take its reign. So Amanita as a genus, and again, this Amanita genus has 600 different species in it, is theorized to evolve about 50 million years ago. What makes Amanita so special and so different than other fungal species is mainly its ability to produce these super potent toxins. And these super potent toxins, and the biggest one specifically, is called alpha amanita. We'll talk about what alpha amanita does to our body in a couple minutes. It's absolutely wicked. But all of these Amanita species have alpha amanita toxin within them. It's just that death cap, what makes them different and how they evolve differently is they have a larger concentration of alpha amanita within them. Another thing that makes Amanita species a little bit different than what we were seeing in the past is their ability to form these symbiotic relationship with trees. When they were evolving, they started to lose the ability to form cell wall digesting enzymes. This posed them a problem because if a fungal species isn't able to digest cell walls, then they can no longer decompose plant species that might be dying around them. So they have to come up with a different way to gain nutrients from their environment. The way that they did this was by forming relationships with nearby trees or nearby plants so that they did not have to decompose to get their nutrients. Instead, they could just swap some nutrients with those plant species. So that's one part of the evolution. We see them losing the ability to form digesting enzymes. And so instead, they're forming these relationships with the plants around them. And they're getting their nutrients from the photosynthesis that the tree is doing. So cool. When they lost the ability to form these cell wall digesting enzymes, they also freed up a lot of energy for their cute little mushroom bodies. So, what are they using that energy for now? To create these highly potent toxins that can kill anything that eats it. Of course, why wouldn't you do that? It's gnarly, it's cool. Of course, Evolutionary biologists are, like, rolling their eyes at me right now because evolution, we're not choosing to evolve in these different cool ways. But the death cap, how it particularly evolved to have this trait of forming alpha amanita toxins is through a process called horizontal gene transfer. Horizontal gene transfer is when organisms acquire genetic information from their environment. So you can imagine it as them engulfing or swallowing up this genetic information that's hanging out in the environment. And this allows them to gain new traits. So it's the leading hypothesis that all Amanita species were able to evolve this ability to produce these toxins by engulfing these traits from their surrounding environment. And now they are by far the deadliest genus of fungi around. So, shout out to them for doing that. Now, these potent toxins, they evolved to have them, but what makes them so beneficial and that now they've stuck around for 50 million years? Because evolution, if we're evolving a trait, it needs to be beneficial to continue to be passed down to further offspring. So, the big benefits come in two different ways. One of them is because it's protecting them from predators. We learned this, it's just an adaptation. We want to be protected from predators no matter what you are. You do not want to get eaten. So, that toxin theoretically is going to protect them from predators because if we have a squirrel who eats these death caps and dies, the squirrel friend that was watching that behavior is probably, hopefully, not going to then go and eat a mushroom that looks like the mushroom that their friend just ate, because guess what? They don't want to die. But let's say that this squirrel friend isn't very smart and goes and eats a death capped mushroom, even though they just saw their friend die. That's still beneficial to the death cap and to all of these other Amanita species who are utilizing similar traits. The reason it's beneficial is because when the squirrel dies, it's dying on the soil. Now, death cap is not actually doing the decomposing of those squirrels because, again, they've lost that ability. They don't want to spend all of the energy on it. However, other decomposers in the area are indeed going to decompose that squirrel. When it's being decomposed, all of the yummy nutrients that are no longer serving the squirrel are going to now be restored and recycled back into the soil. This creates an environment with super nutrient rich soils, which is going to help the tree that the death cap has this lovely relationship with. So the tree is going to be able to take up nutrients from that soil. It's going to be able to photosynthesize better because of it and survive longer because of it. And if that tree is photosynthesizing better and surviving for longer, guess who also is going to thrive? Our death cap. So these toxins not only protect it from predators, but if they happen to get eaten, that fruiting body, that mushroom that was just eaten, is not the life and blood. The life and blood is the mycelium underneath the soil. So really, it's still getting more nutrients and it's getting what it needs to produce even more fruiting bodies and continue its life cycle. I've mentioned a couple of these already, but I wanted to specifically shout out what makes Amanita phalloides so special because we've talked pretty generally about Amanita as a genus, but Amanita phalloides, or our death cap, a couple of things make them very distinct and make them their very own species. For one, the vulva is very distinct. So if we look at vulvas of other Amanita species, these vulvas might hug the stem a little bit closer, but in a death cap, the vulva is going to be very cup-like, and it's going to be a little bit more widespreading and pronounced. Another thing that makes death caps a little bit different is because they do not have any of those warts on top. I want you to picture Toad from Mario. Toad is a little Amanita species, and The Amanita species has those cute little white warts on top that make them very beautiful. Death caps don't have that. Like I mentioned before, their caps are going to be very smooth without any warts on them. And last but not least, it's by far the most toxic, and it's going to be the most far reaching because we've spread it all over the planet now, and it's able to be invasive in lots of different areas, which makes it different, it makes it special, and it has created this scenario that it is so renowned for being so deadly to so many different things. The next step is going to be to talk about where we find this, how it moved all over the world. We'll talk about its life cycle a little bit too. But before we do that, I want to take another break. So please go get a drink of water, pet your dog, and I will be back in a sec. Podcasts are a way for us all to break up the monotony of our day, either while we're taking walks, or we're at the gym, or we're cooking a meal. One of my favorite podcasts to go to is called This Podcast Will Kill You. It's about infectious disease and the healthcare system and the historical preference of pathogens. And it is so interesting and insightful, and I've been an avid listener for years. If you like this podcast, I'm sure you will enjoy This Podcast Will Kill You, and you should find it on any of your podcast platforms. This is in no way a sponsorship, just a nice little shout out to one of my favorite podcasts of all time. This Podcast Will Kill You. Go listen. And we're back. Our next task is to talk about where we can find death cap, where we were finding them 50 million years ago, where we find them today, and then we'll talk about the life cycle of death caps a little bit too. 50 million years ago, when death caps first evolved, we were finding them in places mainly in Europe, and particularly the UK and Ireland. The reason for this is because they evolved to have a relationship with English oaks and beeches, and this relationship served them well, so they continued to evolve with these English oaks and beeches, and that's where they colonized when they were first starting out. They also like these really mild, wet climates. So, those particular areas of the world have temperatures that are not too hot, not too cold, and they're very moist. So, they have lots of precipitation going on to support the mycelium network and to support the trees that the mycelium network is in cahoots with. They're now invasive across the entire planet except for Antarctica. So we need to ask the question, how did they get from just these couple of places in Europe, Ireland, and the UK to all over the planet? My quick answer for you is humans. This really started to boom in the late 19th and early 20th century with increase in global trade and also with our lovely world wars that we had. It's very, very likely that what happened is while people were importing trees, we had some spores coming over that were either being carried on the roots of those trees or carried in the soil that those trees were with. And when said tree gets planted in California, well, bam, now we have death caps in California. And this is how it happened kind of across the world. In the United States, the first recorded sighting of a death cap was in 1938 at Hotel Del Monte in Monterey, California. Then we see them popping up in Australia in the 60s, and it just kind of started snowballing from there. We've seen them all across Asia. We've seen them in Africa and South America. Again, everywhere except for our good friend Antarctica. And good news for the death cap is that they've been able to evolve in these different areas as well. So we're seeing them evolve in different ways across the world now, which does make them problematic for us, but it's really, really cool for them. I'm going to take us down this little rabbit hole of how they're evolving in different ways, but I'm just going to do it through the lens of how they reproduce, which kind of gets us into the life cycle of these death caps. Death caps can reproduce asexually or sexually. However, 50 million years ago, when they were colonizing those places in Europe, they just were producing sexually. Now that they've moved across the world, there's booming research coming out of California about how these death caps now have the ability to reproduce asexually as well. I am going to take us down both of these avenues. First, we'll do sexual reproduction, then we'll do asexual reproduction. And that will allow us to meet in the middle to talk about the rest of their life cycle. Biology 101. In sexual reproduction, this is when two mycelium networks meet underground, and these mycelium networks are genetically different from one another. When they meet, what is going to happen is a fruiting body is going to come up, aka a mushroom. So these two mycelium meet underground, then they form this mushroom. The mushroom will grow, it will turn into a mature mushroom, and particularly because this is sexual reproduction, the spores that are being created in the reproduction process are going to go through a process called meiosis. This process is really, really important to sexual reproduction because what happens is cells from one parent meet up with cells from a different parent, and those cells, while they're replicating, they're going to undergo an event called crossing over. What crossing over is when the DNA of those cells swap. They're crossing over, they're swapping information, they're mixing up a little bit. So now the spores that have been created in this new baby mushroom are going to be genetically unique than either of the parents that we started off with. This is how reproduction happens in humans. We have this process of meiosis that's happening in sperm and egg so that the genetic information is, mixing up so that we don't have clones as humans. Mushrooms do this too, the death cap specifically. So the original species that we had 50 million years ago underwent this process of sexual reproduction. We had all of these genetically diverse mushrooms, and those spores would then catch the wind. They would go and travel to different places, and we have this lovely diverse group of death cap that are surrounding the tree. Now in California, when we brought them over through that international trade, these death caps in California began to evolve so that they were able to undergo asexual reproduction. This process is pretty similar, but there's one key difference, and that key difference is from a mycelium network, we can have fruiting bodies pop up on their own. So we'll have a mushroom pop up, and while that mushroom is creating spores, those spores, instead of undergoing meiosis, they undergo what's called mitosis, which sounds really similar, but they're different. And the big difference is that instead of that crossing over event happening where we're mixing up the DNA, we're just going to make exact replica copies of the parent DNA. So we'll have spores with the same exact genetic information that will then be dispersed in the wind, go in, find some nutrient-rich soil, and we'll have a mushroom that is genetically identical to the mushroom that it came from, which can be really beneficial. The reason that this is really beneficial is because in areas where we don't already have a lot of mycelium network or a lot of mushrooms, that single mycelium network is no longer required to find a genetically unique partner to mate with. They can reproduce all by themselves. So it lends them to be able to colonize areas a bit quicker and a bit easier because they don't have to find these partners. They do not have to have another mycelium network to reproduce with. If we then think about what happens after these spores hit this nutrient-rich soil. We have these spores dispersing in the wind, and then they will go and find nutrient-rich soil, hopefully by a tree, and things start to happen pretty quickly from here. The spores will create new mycelium network. They're going to divide and divide and divide. So we have these lovely hyphae and the nodes, and they're finding trees to swap nutrients with. And the way that we get a fruiting body is really all about environmental conditions. When there has been a lot of rain, when it's nice and warm outside, we're going to have these little pins start to pop up. The pins are the babiest of baby mushrooms that you could possibly think of. They are so cute. And these pins are going to pop up out of the ground. And now, after this pin has popped out of the ground, over the next 3 to 7 days, that little pin will turn into an adult death cap. These adult death caps have all of those structures that we talked about at the very beginning, including, most importantly, the gills and the spores within the gills. Around that 7 day mark, those spores are going to be ready for dispersal. So this is happening very fast. They're only alive for a week, and then they're going to disperse all of these spores around the 7 to 10 day mark. And after those spores are released, that fruiting body, that death cap mushroom, is going to rapidly decay. It's going to go back into the earth. All the nutrients that were within it are going to be decomposed, but the toxins don't decompose with it. So please also don't eat a decaying death cap. Don't try it. It's going to end up very bad for you. As that mushroom is decaying, what the coolest part is that the mycelium network underground is alive and well the entire time. It can live for decades. I saw estimates for up to like 30 years. So there are probably mycelium network in my yard that are older than I am, and that is crazy. But the mycelium network keeps doing its thing, and it's going to continue to produce these fruiting bodies when the conditions are just right, so that those death cap mushrooms can then produce millions, sometimes billions of spores, so that they can be dispersed and then they can decay, and the process just starts all over again. I'm going to do a quick side note here about the decayal process of these death caps or mushrooms in general, because I found this really cool paper that I want to shout out. And I'll have this paper in my sources if you want to give it a look. But when we're thinking about how a mushroom is decaying, we're thinking about all of the water that's inside of that mushroom being evaporated. And I want you to remember that these spores that are coming out of the gills have to be dispersed. And usually that dispersal is going to happen through wind. But even in areas that are not very windy, or if there isn't a lot of wind in a particular area at a particular time, as water is being evaporated out of that mushroom, that evaporation event is actually creating this really slow airflow, and that airflow is then dispersing the spores. It can disperse the spores up to 10 centimeters in length, which is crazy because those spores alone are super microscopic. And the airflow being created by this water is just going to carry them away, which is one reason that mushrooms need so much water, but also a reason that they decay so fast is all of this water is going to be evaporated out of the fruiting body really quickly, so that it's creating this airflow so that spores can move with the airflow, so that that life cycle can start again, which I think is absolutely fascinating and such a cool evolutionary little niche that they have. It's so cool. But all of that to say, they're very evolved for their particular areas. And as we continue to research how they're evolving in different areas of the planet, I'm sure that we will discover new little knacks and treasures that these death caps have, and maybe one day they might even create a new species in and of itself, just based on how they've been dispersed all over the world. Our last step is going to be to talk specifically about how death caps kill people, which I'm so excited about. But we're going to take one more break before we do so. Go grab a snack, drink some water, stay hydrated. We'll be back soon. If you like a good mix between thriller, historical fiction, and current events, I have the perfect book for you. Yesteryear by Carol Claire Burke focuses on a protagonist living a traditional farming lifestyle, but this traditional lifestyle might not be as clear as it seems. It had me on the edge of my seat and gave a good look into history while also highlighting current societal themes. If this seems like a story for you, find it at your local bookstore or library. This is in no way a sponsorship, but I hope you find as much joy in reading it as I did. Now, let's get back to the show. Our very last stop of the day. How do death caps kill people? I'm so excited to tell you. If you'll recall, that toxin that death caps evolved to have, alpha amanita, it really is the star of the show. If we think about how much amanita is required to kill a person, specifically if we think about how much death cap is required to kill a person, the lethal dose is going to be about 5 to 7 milligrams of that alpha amanita toxin, which can be in just a single death cap mushroom. The lethal dose of death cap is probably a 20-gram mushroom, give or take. In that 20-gram mushroom, we will have around those 5 to 7 milligrams of alpha amanita. This is a big generalization because, of course, this is going to depend on your body size. It's going to depend on also if your dog eats it versus if you eat it. But if you are eating death cap, if you eat a single mushroom, you could and probably will die. So please don't do that. But we're going to set the scene. I'm going to tell you how it's going to kill you. So you've gone out into the forest, you took a couple of friends, and you thought that it would be so cute and whimsical to go and forage your own mushrooms, even though you don't know very much about it. What's the worst that could happen? So you and your friends are foraging mushrooms and you're going to cook them into a meal that you're having tonight. You go back home, you cut up these mushrooms. They look exactly like the mushrooms that you get from the grocery store. And also, when you cook them, they smell delicious. And when you eat them, they are delicious. And then after your meal, you're feeling great. You just shared a meal that you foraged yourself from the woods right next to your house. And you had that meal with two lovely friends. Again, what could go wrong? Now, fast forward 6 to 12 hours later. That meal that you had is giving you a bit of a tummy ache. The first symptoms that we're going to see are almost always going to be gastrointestinal. This is depending on how much of that alpha amanita toxin you actually consumed, but typically we're going to start with some diarrhea, some vomiting, about 6 to 12 hours after the meal that you have. Along with the vomiting and the diarrhea, you might have some blood in your stool, depending on how much of the toxin you did consume. However, if it's just some diarrhea, just some vomiting, you probably are not going to rush to the hospital because these are symptoms that have come up time and time again in your life. Hopefully, not too often. If it is that often, maybe you should go see a doctor. But you've thrown up before. You've had diarrhea before. It is probably just a small bout of food poisoning. No reason to worry quite yet. Now, after you've thrown up a little bit, you've had some diarrhea, you might enter this stage of what we call latency. For the next 12 hours, up to the 24 to 36-hour mark, you'll probably be feeling pretty okay. You might still have some diarrhea, but nothing horribly bad. But when you get to that 3-day mark, maybe 72 hours, maybe even 60, you're starting to feel absolutely dismal. You might be getting a little bit jaundice. You might be completely lethargic, and then you choose to go to the hospital. Once you're at the hospital, they run some tests, and what they see is that your liver values are absolutely shot. Your liver is in complete failure, and when they start asking you questions about what you've been doing over the past couple days, you tell them that you went out foraging in the woods and that you foraged these mushrooms that look like the mushrooms you get from the grocery store, and you cooked them in your meal. And then they say, "Oh no, you have death cap poisoning. You're going to die." But let's back up. Okay, so what is it doing to your body, right? Of course, it's causing those gastro symptoms because as soon as alpha amanita is hitting your intestines, is hitting your stomach, it's going to be wreaking havoc on your cells. What it's doing to those cells is it's completely inhibiting the function of a super important enzyme called RNA polymerase 2. So it's basically killing your cells right off the bat because what RNA polymerase 2 does is it helps our cells synthesize proteins. Proteins are in charge of so much in your body. They're giving the code for your genetic material, and they're also helping with biochemical reactions that are happening at any given moment in your body. So without proteins, your cells are going to die. So it's doing that to your digestive system first, right? Your digestive system is saying, something is wrong. But as your digestive system is continuing to do its job, it's going to be absorbing all of the food that you just ate, all of the nutrients that you just ate. That absorption process then sends waste products to your liver. One of those waste products is unfortunately alpha amanita. Once that alpha amanita reaches your liver, the alpha amanita is going to do the same exact thing to your liver cells. It's going to inhibit the function of RNA polymerase 2, and it's going to start quickly killing those cells because, again, without RNA polymerase 2, we don't have proteins, and without proteins, your cells are dying. And it's happening really rapidly. Like I said, at about that 3-day mark, once you're feeling absolutely dismal and you go to the hospital, there's nothing that they can do for you because so many cells have already died in your liver, which is very, very frightening. And especially so because these symptoms are so sneaky. Like, as we were talking through this story, of course, you're not going to rush to the emergency room because of diarrhea and because of vomiting. Of course you're not, especially with the healthcare system here in the United States. But because you waited, now medical professionals can't do anything for you. Technically, there are antidotes that we could give for the toxin alpha amanita. I don't know a lot about them because that's kind of above my pay grade, but those toxins or those antidotes can help reverse the side effects of alpha amanita, but particularly just within the first 24 hours. So, if you're administered those antidotes within the first 24 hours after your meal, their survival rate is up to 90%. But let's break that down a bit because if you're standing in a lineup of 10 people, that means the mortality rate is still at a 10%. So if they give you all a death cap to eat and they say, but here in 24 hours, we'll rush all of you to the hospital, no worries. Well, guess what? One of you is still going to die, and I wouldn't want to be in that lineup. But if we wait until after the 60-hour mark, so anywhere between like 24 hours to 72 hours, the mortality rate starts to spike pretty quickly. If we wait 36 to 48 hours, the mortality rate is now at a 50%. If we wait the 60 to 72-hour mark, it's now like at a 75%. And anything more than that is going to be a 90% mortality rate. It's really, really deadly, like crazily so. Lots of humans have died because of death cap poisoning throughout our time on Earth. I wanted to mention just a few that stand out and that are a little bit fun. So we're going to go on a little bit of a history ride here. The most notorious and the earliest recorded comes from 54 CE, and this was in ancient Rome, where Emperor Claudius was poisoned by his wife by death cap, and that Emperor Claudius ended up dying so that her son Nero could secure the ancient Roman throne in 54 CE. We also have the death of Holy Roman Emperor Charles IV in 1740. who was poisoned by death cap, and that started the War of the Austrian Succession, which I don't know a lot about, but seems like a really big deal. And then we also have a famous German composer from 1776 called Johann Schobert, who took his whole family and some friends and a servant out to the woods despite advice from a couple of local chefs that said, hey, you shouldn't go and forage when you don't know what you're doing. But he did so anyway, and the group foraged some death cap mushrooms and they all ended up dying from them. So, all of these stories and so much more led the death cap to become the death cap. In 1871, it got coined its name, the death cap, by Mordecai Kubik Cook in his Handbook of British Fungi. He was a renowned mycologist, and he had all of these stories about death cap poisoning and accidental deaths from death cap. And he said, this one's pretty deadly. We're going to go ahead and coin it the death mushroom. And shout out to him for that because I love the name death cap. And even though it has this name death cap, I wanted to emphasize that while it does account for 90% of mushroom-related fatalities, and it can be this scary guy, it still only kills about 100 people per year. It's more people than should be dying by mushroom poisoning, but there are far better things to be afraid of. Like, maybe the American healthcare system. But I digress. Far better things to be afraid of. Don't be afraid of death caps. And if you want some whole picture advice on how to not get death cap poisoning, here's my advice for you. For one, don't go foraging in the woods unless you are an expert or you take an expert. And if you do neither of those things, please do not go and forage because it could end badly for you. My second piece of advice is if you're like me and you get a little stressy messy about health things, if you go to the grocery store or if you go to your local farmer's market and you buy mushrooms and you cook those mushrooms in a lovely meal for yourself and you have a tummy ache about 6 hours later, I am nearly certain that you did not eat death cap. So please do not let this episode frighten you or make you anxious. And please, for the love of all good things, do not go to Google and doom scroll death cap poisoning because it's just going to make you feel worse coming from your resident doom scroller. My last and biggest piece of advice is simply respect the fungi and they will respect you. I don't think that's my best advice, but it does add some whimsy to my day, and I sure hope it adds some whimsy to yours. And that, my friends, brings us to the very end of all of the information that I have for you about death cap, also known as Amanita phalloides. Before we officially wrap up and I give you a couple of homework assignments, I wanted to go to Midnight Magic and I wanted to see where we'll be headed here in a couple weeks. So let's go ahead and do that together. Let's see what they want from us. Okay, here we have the Two of Swords, which is standing for our Piggyback Rosegill. It's also known as Borrelia surrecta. Look at that pronunciation, did real well. And I'll be telling you all about that here in a couple weeks. I'll also be posting this on our socials. And here is where your homework assignment comes in to please follow our new shiny bright Instagram and TikTok accounts. Again, it is at the Fungi Oracles, all lowercase. There I'll be posting the picture of this card that I just drew, the Two of Swords for our Piggyback Rosegill. I'll also have different resources that you can reach to on there. So both on our Instagram and TikTok, you'll find a link tree. And in that link tree, what you'll find is sources for today's episode that you can look at and read more about if you'd like to. You'll also find a transcript of today's episode, so you can do that reading if you'd like to. And then last but not least, what I'm most excited about is you'll see a link for what I'm going to coin as our Fungi Fridays. These Fungi Fridays are meant for throughout your week. I would like for you, while you're out on your walks, while you're out on your jogs, to snap any pictures that you might see of any fungi that you come across. And on Fridays, what we're going to do is we are going to post those. So hopefully we can, throughout our years together, be posting fungi from across the planet, and we can all look at them and admire them together as fungi lovers. All of this will hopefully be in a website one day, but for now, what I have for you is a link tree. So please go to those socials at the Fungi Oracles, look through those resources, give me pictures of the fungus that you come across, of the mushrooms that you come across, so that we can all share in this love together. Thank you so, so much for being here with me today for our very first episode. It means the absolute world, and I cannot describe how excited I am to continue to go on this journey. And here in a couple weeks, we have the Piggyback Rosegill to look forward to. I love y'all. Thank y'all. Have a lovely rest of your day, and I'll see you in 2 weeks.